The field of spatially resolved transcriptomics has demonstrated how science can illuminate the hidden complexities of our world by mapping the intricate landscape of gene expression in each tissue or organ. Non-content with only measuring the transcriptome, spatial biology has extended to cover a multitude of modalities. From chromatin accessibility to the proteome, we now have an unprecedented ability to explore the mysteries of development and disease. Yet for all its power, spatial biology remains a relatively new field, and computational tools to analyze this data are required to capitalize on the wealth of information that is generated. We developed Vesalius, a computational tool that can effectively recover tissue structure in multiple spatial biology modalities. At its core, Vesalius converts spatial biology assays into grayscale images upon which image processing techniques can be applied. The initial step necessitates the pre-processing of spatial data, wherein lies the task of normalizing the data and extracting variable features. Subsequently, Vesalius, in its pursuit of reducing the dimensionality of spatial biology datasets, employs an assortment of methods such as principal component analysis, non-negative matrix factorization, or latent semantic indexing. In parallel, punctual spatial indices undergo an expansion, metamorphosing into pixel tiles courtesy of Voronoi tessellation. For each latent space dimension, Vesalius assigns to a tile its respective embedding value to generate a grayscale image stack. We have reconfigured image processing tools, ensuring their seamless functionality on images of higher dimensions, what we aptly refer to as image tensors. The selection of tissue territories occurs by segmenting this image tensor, followed by further subdivision of chromatic segments. This process ensures that territories are spatially distinct even if they belong to the same color segment. Interestingly, we found that visual inspection of latent space embeddings as grayscale images provides a unique and powerful way of selecting dimensions for further analysis. In some instances, spatial territories that are often missed by other methods lurk in lower variance principal components. As an example, we ran Vesalius on the newly published StereoSeq datasets that provide subcellular resolution, spatially resolved transcriptomics of the mouse embryo. We were able to recover many tissue territories, including different brain sections, the spine, the liver, and the heart. The very essence of Vesalius lies within its versatile nature, granting us the unparalleled opportunity for effortless multimodal analysis. With little modification, Vesalius is able to generate latent spaces for multi-omic measures that can be then converted into an image tensor. Recently, excellent spatial site-seq datasets were released to the public. This technique allows for the joint measurement of RNA and protein at the same spatial index. We can easily recover the tissue territories that summarize gene and protein expression profiles in mouse kidney. We can go a step further and isolate territories characterized by unique expression of both mRNA and proteins. Vesalius generates a common embedding that will be used to create grayscale image tensors. As long as you can generate a multi-dimensional latent space, Vesalius can convert it into an image tensor. A final challenge that we have attempted to address with Vesalius is the one of spatial alignment. Given two spatial datasets that can differ in orientation, structure, and transcriptional profile, can we align one onto the other such as that both the transcriptome and spatial context are matched between datasets? Vesalius attempts to solve this by considering the alignment problem as similar in nature as an assignment problem, where the goal is to assign workers to tasks that would reduce the overall cost. We build a cost matrix that considers transcriptional distances between two cells, but also the transcriptional distances between each cell's spatial niche. The smaller the distance between two cells, and their niches, the more likely they will be paired together. To test this, we selected a small sample of a slide-seq dataset in the mouse brain as a reference. We created our query by flipping the coordinates, adding noise to the coordinates and the counts, and randomly selecting a subset of these points. Our initial trial demonstrates that Vesalius can accurately map the query dataset onto our reference dataset and recover the expected territories. While there is progress to be made in terms of runtime and software, we hope to have demonstrated that the latest version of Vesalius provides a flexible and visual way to analyze multi-omics datasets. In the future, we hope to provide a robust, high-performance and interoperable tool to complement your spatial analysis pipelines.